Today's topic, the DNA replication fork. To begin, let's start with our original DNA strand. Our origin of replication here is going to begin on the left side of the DNA strand, colored in red. As DNA replication begins, there are a few important enzymes to discuss. The first is gyrase. This enzyme relieves supercoiling before the replication fork, allowing helicase to break the hydrogen bonds in the DNA to separate it into two single strands. This allows one original strand of DNA to be made into two identical copies. After the unzipping begins, an enzyme called RNA Primase comes in. This enzyme synthesizes a primer composed of a short strand of DNA. In blue, you can see what is known as single-strand binding proteins. These proteins bind to DNA to protect it, stabilize it, and prevent hairpins. The priming is critical, as DNA nucleotides can only attach to existing strands, so we need this primase to give us a place to start. It is critical to note that DNA is created from the 5' to the 3' end, explaining why the RNA primer is located in these spots. Once the primer is in place, DNA Polymerase III comes in and adds DNA nucleotides to the primer. Remember, this is always done in the 5' to 3' direction. As the 5' phosphate group of a free nucleotide attaches to the 3' OH group of the last nucleotide in the strand, two phosphates are removed, forming a phosphodiester bond between the two nucleotides. As Gyrase and Helicase work together to unzip the DNA, this process continues. As you can see here, the bottom strand requires another RNA primer in order to continue working in the 5' to 3' direction. As you can see, more and more nucleotides are added to the strands. Because DNA is antiparallel and replicated only in the 5' to 3' direction, you can see that the strands on the top and bottom look a little bit different. The top strand is known as the leading strand and has continuous DNA replication, hence no breaks in the strand. On the other hand, the bottom strand has these nicks in the strands. These nicks separate what is known as Okazaki Fragments, and these fragments are caused by discontinuous DNA replication, something that occurs in the lagging strand. As this process continues, you can see that these nicks will remain and the RNA primers. This is where DNA Polymerase I comes in. The enzyme converts the purple RNA into DNA. Now we have one last step. DNA Ligase comes in to seal the nicks that were created in the lagging strand that separated the Okazaki fragments. Once this is done, you can see that the DNA on both strands is continuous and complete. This whole process continues until the entire original strand has been separated and replicated. In the end, we end up with two strands of DNA that recoils back into its double-helix shape. The two strands are identical copies of the original strand, and they both maintain the antiparallel structure of the original strand based on all these components working together.